A parasite is pretty cool. It's pretty good at being able to live in a host for a very long time, and I've got to give it some credit for that.
Hello, this is Jeffrey Fox hosting the August 2016 episode of the Microbe Podcast. In this interview, I speak with Emma H. Wilson of the University of California, Riverside. First, let me remind listeners that I welcome feedback. Please email me via jfox@asmusa.org or contact me on Twitter @JeffNF. During this podcast, I speak with Wilson about recent research from her and her collaborators revealing how Toxoplasma gondii parasites directly disrupt the neurotransmitter glutamate in the brains of rodents such as mice that they infect, leading that neurotransmitter to build up and thus disrupt the behavior of the animals. A brief article describing some of this work appears in the August 2016 Microbe. For details, go to asm.org. I think it would be helpful if you would summarize your recent findings with Toxoplasma gondii and the way it's impinging on the metabolism of the neurotransmitter glutamate, and then we can get into the details and the ramifications of that work. But I'd rather you put down the predicate and summarize it than I try to.
Okay, well, I think the lead up to this study was that Toxoplasma is a protozoa that causes a chronic infection, is very common throughout the world. In the human population, 30% of people are infected. And there has been a growing body of correlative studies suggesting that the location of the parasite in the brain and the chronicity of this infection could manipulate host behavior or also lead to neurological diseases. And there's correlations with schizophrenia, weaker ones with Alzheimer's disease and epilepsy. And these are all quite diverse diseases with sometimes quite unknown etiology. And we wanted to try and look a little more neurologically at what was going on the brain of an infected mouse. These studies are not possible in a human. And one of the observations that has been present in the literature for a very long time is the activation of glial cells called astrocytes. And they get highly activated and swollen. And when these cells become so activated, they are unable to do their normal physiological job of helping neurons. And one of the prime things that they have to do is to soak up the neurotransmitter glutamate. So after a neuron fires, it releases glutamate. And if that glutamate in the synapse isn't cleared away, then that noise to signal ratio is lost and neuronal circuits fail to work properly. So astrocytes, these glial cells, soak up the glutamate via a transporter called GLT1. And what we discovered in this paper is that infection with Toxoplasma leads to a downregulation of this transporter. And as a result, we see significant, massive increases in this neurotransmitter glutamate. And that leads to neuronal pathology. One of the reasons we think this is particular to this transporter GLT1 is because we can rescue a lot of this pathology by treating our infected mice with a beta-lactam antibiotic called Ceftriaxone. And this specifically upregulates the transporter. And we measured some of these pathologies and effects looking at using microdialysis of the fluid and extracellular space. So we measured all those amino acids, including glutamate. And we also did some EEG analysis, the same type of EEG analysis that would be done on an epileptic patient, for example. And we can measure the neuronal firing that is occurring in infected mice and compared to uninfected and compared to our treated, rescued Ceftriaxone treated mice. So along with other recent papers, this is starting to get at the actual neurological mechanisms that could be responsible for any changes in behavior or any of these correlations with neurological diseases.
And in this case, are the mice that you've been looking at, are they showing other hallmarks of epilepsy? Or is it just in an electroencephalogram pattern that corresponds to epilepsy?
Well, for those of us who have dealt with these infected mice for a number of years, we have now and again seen mice having seizures. And that's not really been quantified before. A colleague of mine, Ira Blader, has also published a paper demonstrating their susceptibility to seizures. So when you actually induce seizures chemically, you can see that mice that are infected with Toxoplasma have a lower threshold of seizure. So they're more likely to have seizures than uninfected mice. Although if we look at our mice side by side with uninfected mice, then you couldn't tell which one is infected. There's no outward signs of disease. Then it seems that these mice are also susceptible to seizures.
Okay, but the EEG patterning, the electroencephalogram patterning is very readily distinguished.
Yes.
I'm going to infer, or ask.
Yes, my colleague Devin Binder, who is also on this paper and helped us do these studies, he's an expert in epilepsy and is actually a neurosurgeon on Tuesday, Wednesday, and Thursday, and runs his lab Monday and Friday.
I see.
So he knows these patterns, and straight away was amazed at the difference in the infected mice. So you lose the synchrony of neuronal firing, and there's a massive drop in the power of these neuronal circuits. So it's very clear.
Okay. Let me step back a bit. I did want to, it's important to establish that the parasites operate in two distinct phases, right? In the central nervous system, some of the time they can cause an active fulminant infection where they're affecting these activities, the firing of neurons and all the other things that go on in the brain. And at other times they're there in a latent state. And as I understand it, they really aren't doing much of anything to the brain when they're there. They're just waiting for an opportunity, or do they have some activity and some impact on the physiology of the central nervous system when they're in the cyst state?
Yes, I think for a long time we've thought of chronic toxoplasma and the cyst that is located inside neurons as almost like a latent virus, that it's really just sitting there, and as soon as you lose your immune response, which is seen in AIDS patients, for example, or severe chemotherapy or transplantation, then you will get reactivation of those cysts, and that's when you get real disease. But I think we're starting to veer away from that a little bit, that it's not as latent as we really thought, and we see changes in cyst burden. And another colleague, Tony Sinai, has shown that there's big changes in cysts, that the parasites are growing and replicating inside these cysts continuously, much more so than we previously took for granted. So there's a lot more activity, and then this is a cyst that is located intracellularly within the neuron, and our understanding of how that changes neuronal signaling, these are very polarized cells, so the cell depends totally on being able to signal from one end of the cell to the other. And meanwhile, you've got a cyst that is inside it, which is, can be up to 100 microns. It's really 12-month pregnant neuron, and how that could affect those few neurons, those relatively few neurons that are infected, how that changes neuronal signaling and firing and behavior has barely been touched upon at this point.
Okay, so there's more activity at the cyst stage in itself, and it seems that as a cyst grows, it could be a Trojan horse or something. It could be releasing, it certainly must be releasing any of a number of biochemical materials that could affect what's going on within a neuron.
Yes.
Okay.
And Toxoplasma is well known for being able to manipulate the host cell. It can turn off important signaling pathways that are responsible or required for the pro-inflammatory immune response that controls it. So it turns off signaling pathways like NF-kappa B, manipulates STAT1 signaling, and all of these pathways are responsible for the cytokines like IL-12 and interferon gamma that ultimately are able to control protozoan parasites. So,
okay, but it sounds like, I don't know, maybe I misunderstood you, but it sounds like now you're again talking about the active stage of the parasite rather than the cyst stage, or are you?
Yes.
What you've just said applies to both stages.
Well, I suppose all that work, all that cell manipulation work has been done in investigating the tachyzoite, the fast replicating active stage, but we have no idea what this bradyzoite, this cyst, could also be doing. And I believe that it's probable that there are similar abilities for the bradyzoite and the cyst to manipulate the cell that it's in.
All right, so it's maybe it's not as loud in a sense. Because it's slower growing and it's more isolated because it's partly encapsulated, right? But it still might be doing similar things to the active state parasite.
Yes. Yeah.
Okay. Now, the effects that you began by talking about, the effects on glutamate, that's occurring. Well, I guess I better be sure which cell the Toxoplasma is in. Is it in the astrocyte, or is it external, or is it in the neurotransmitter cells? And if that's the case, is it confined to glutamate type neurotransmitters? Because there are plenty of other, or at least, my knowledge of the central nervous system is not so up to date, but I know there are at least a half dozen, say, neurotransmitters that different chemistry than glutamate.
Yes.
Bombarded you with a bunch of interlocking questions, but maybe you can make better sense of it than I could of the question.
It's lots of great questions, and I think we have to work them all out. There is absolutely. So again, my colleague Ira Blader has demonstrated that GABA seems to be dysregulated, or at least the GABA receptors are dysregulated. So that is a neurotransmitter that is responsible for the negative firing of neurons. So that would also suggest that too much kind of positive firing when you have too much glutamate and too little negative firing due to GABA, then we're definitely going to be more susceptible to seizures. Where the parasite is located is a really great question. So initially, the parasite has to come in across the blood-brain barrier somehow, and several people in the field are looking at how that happens and what cells they invade. And recent work by Anita Koshi has generated very cool parasites where they secrete Cre recombinase. So any cell that has been infected can be lit up in the correct mouse system, can be lit up by this secretion of Cre recombinase, so you can track where the parasite has been. And surprisingly, even though astrocytes make up a major portion of the blood-brain barrier, they don't seem to get directly infected. and that the parasite heads straight towards neurons. And if that's the case, we still need to understand what is that initial signal that downregulates the glutamate transporter GLT1. Is it an immune signal? So there's lots of inflammatory components that could be doing this. Or is it the location of the parasite in the neuron that is also... Neurons and astrocytes talk to each other all the time. In fact, astrocytes will not express the transporter GLT1 in the absence of neurons. So there's a constant communication between these cells. So perhaps the location of the parasite inside the neuron is actually indirectly responsible for the downregulation of GLT1. Does that help answer some of your questions?
Oh, it does.
Yeah.
It does. And it just, of course, it also helps to show in Technicolor just how complicated the central nervous system is, and of course its response to the parasite is likely to reflect that complexity. That's part of what you're telling me, I think. My sense of the way that neurotransmitters work is that there's a gap across the synapse, and the neurotransmitter is released from one cell and then hits the other cell across that gap, and there's a receptor there that then responds to the glutamate, and the signal goes on down that cell, and so on and so forth.
Yes.
Now, I didn't appreciate the role of the astrocytes, but they seem to be also part of, at least on the external side of the synapse, are also dealing with the glutamate that has done its task across the synaptic gap. So can you help me out here? Is that picture more or less correct? But it seems as if the astrocyte is serving a secondary role. It's not transmitting nerve impulses, but it is helping to clean up after a nerve impulse has passed by. And is that its primary role here?
Yes.
And that's what's being deranged by the parasite.
Yes, exactly. I think neuroscience used to be people that studied neurons, and they did so in isolation, and neurons are the most important and unique cell of the brain. And actually, for the past probably 20 years, people have recognized that, probably more than that, actually a lot more than that, the importance of glial cells, astrocytes and microglia, but astrocytes in particular, and they're very important in neuronal development and growth and firing, and all that physiology is actually dependent on a very close relationship between neurons and astrocytes. There's a really good book by Doug Feinstein, who has written a lot about glia, and if anyone's interested, they should. I think it's called the Second Brain, or I forget, but it's explains the real necessity of these cells to any of the functions of neurons. So the astrocytes have to clear these neurotransmitters, convert glutamate back into something more useful for the neuron and less toxic for the neuron, and they do that. They're glutamine synthetase, and so they convert glutamate back into glutamine, release it back to the neuron, that the neuron can then safely use that as energy and as a way to release back into glutamate.
All right, so it is secondary to the nerve impulse firing of the glutamate neurocircuits that we're talking about. That's what I'm trying to understand. That you're certainly not positing that the parasite interferes with neurotransmission front and center. It's a secondary effect, but it has the unhappy effect of interfering with the glutamate transmitter pathway.
Yes.
But at a secondary level.
Right. The studies that need to be conducted to test whether the parasite can also directly change neuronal transmission. Those have not necessarily been done. So you would have to do some electrophysiology on infected versus uninfected neurons and see if their function or their physiology is different. So I think those studies will be important ones to do. But in addition, we do know that these astrocytes, it's actually called a kind of tripartite synapse. It's not just the two neuronal cells that are there. The astrocyte is wrapping right around this synapse and is responsible and controls that signal by soaking up the neurotransmitter afterwards.
Okay. I wanted to press you on that because it's really a different picture, and it's fascinating, further fascinating that the parasite, if I understand what you said earlier correctly, is in the neuron itself. It's not in the astrocyte. So however it's interfering with that uptake of glutamate by the astrocyte, it's doing it indirectly. Is that right?
Yes, we think so. Yes.
It's not external to both of them.
Right. That could be one way that it's doing that. It could also be, so this occurs in other neurological diseases where clearly Toxoplasma is not involved. So any inflammation that is in the brain could also result in this downregulation of GLT1. So we do have to define what is that signal that is responsible for the changes in the transporter expression. And that could be a direct result of parasite infection, either the one or two astrocytes that may get infected that then causes some sort of cascade. We do see that GLT1 loss occurs in patches in the brain. It's not a downregulation throughout the whole brain. So the idea that there is loss in patches suggests that there is one particular trigger which would sort of point us towards the parasite. But then the other, as you say, the parasite is predominantly located, nearly always located inside neurons. And again, neurons and astrocytes talk to each other all the time. One of the ways they do that is through exosomes. So the release of these exosomes could be communicating. So the release of exosomes from an infected neuron may be quite different from those that are released from an uninfected neuron. So that might be one way that astrocytes are being communicated by infected neurons. So yeah, all of those studies have to be done.
All right. And yet another part of the complexity is the assortment of host immune responses, which are all taking place on the external side of this set of cells.
Exactly, yeah. And inflammation is a consistent thing during neurological disease. And so when we see GLT1 downregulation in other diseases, this was the use of Ceftriaxone was originally done by Jeff Rothstein at Johns Hopkins, looking for FDA-approved drugs that would
have secondary effects.
Exactly, yeah. So these are drugs that are already approved for use in humans. So he screened a whole bunch of these and found that Ceftriaxone was able to specifically upregulate GLT1. And that actually went to clinical trials for ALS. And perhaps that was, unfortunately, they failed not too long ago. And perhaps it was just too high a bar to meet. But inflammation that is associated with Toxoplasma, there's also components of that same inflammation where we see GLT1 downregulation in other diseases. So although we don't see downregulation of GLT1 and increases in glutamate until almost like at chronic Toxoplasma, chronic infection stage, then whereas the inflammation is actually you get systemic levels of pro-inflammatory cytokines as early as day 3, day 5, and peak kind of around day 7 post-infection. And the inflammation in the actual CNS times very nicely with when we see downregulation of GLT1. So there is a likely immune component to this as well.
Well, let me introduce another, just a sort of freewheeling here, but I am curious because we're talking about a beta-lactam antibiotic, and people are so interested in what the microbiome might be doing. Could some of its effects be indirect in terms of changing populations of bacteria within the microbiome, because it is a potent antibiotic that could very much change what's going on in the gut of the mice you're using, and therefore maybe have an impact on what's going on?
Yes.
Is that worth considering? It could be just a confounding factor if you look at it that way. And I'm not questioning that it has a direct effect on that receptor in the central nervous system, but it could be doing things distal to the brain and yet, having some kind of long-term effect, or not long-term, but distant effect.
Yes, I think we have to consider the microbiome in everything we do. And we did do some kind of investigations into what ceftriaxone could be changing. We did not address the microbiome. We did look in different types of mice to make sure it wasn't just one strain of mouse in which this was going on. So we looked at more resistant mice, and this was consistent within them. So the microbiome, especially when we're dealing with a really solid antibiotic, could easily be involved. We did look at how it changes the inflammatory component of the brain, and we looked at parasite burden in the brain to see if ceftriaxone changed any of those parameters. And there was no change in parasite burden, and there was no change in the level of immune cell infiltration into the brain. I think those studies, the microbiome studies, would be important to understand the mechanism of ceftriaxone controlling GLT1 in the first place.
You described some of the impact of the parasite on the mouse is to give it a brain pattern that looks just like epilepsy.
Oh, yes, yes.
And I was asking whether the effects you see also, are there any signals that resemble other central nervous system diseases by what you're now seeing at the biochemical neurotransmitter level has to do with derangement of glutamate activity? Yeah,
I don't know if we have enough information about that, and if we have enough information on the EEG of signals that are in other patients. So that still needs to get worked out. But one of the questions I had when we were starting this study is, well, how can one infection be correlated with such a diverse number of quite different neurological diseases? And I think a disruption of the primary neurotransmitter glutamate could explain that, because if you have disruption depending on where the parasite is in the brain, you could have sort of very localized neuronal dysfunction, and that might end up triggering many other different types of disease.
I see. So if the focal point, if the main concentration of the parasite is, I don't know, in the forebrain versus the central brain or something, I'm sure that's the most unsophisticated way of putting it, but that would determine what's really what the syndrome that develops.
Yes. And if we tentatively kind of extrapolate what could be happening in a human brain, which we've absolutely no idea about, if you think of a mouse brain has a maximum in these circumstances of maybe 3,000 cysts. So that's 3,000 neurons that are infected. And in a human brain, the ratio of the number of infected neurons to uninfected is presumably far greater. So the location and the loss of GLT1 may be very important in the disease outcome. The other thing to mention is that children who have congenital infection are very susceptible to seizures. And this is one possible kind of wonderful outcome about this study, is that a colleague, Rima McLeod at Chicago, is kind of the expert on congenital Toxoplasma infection. She's very interested in wondering if whether we can treat some of these children, these very young kids that have kind of uncontrolled seizures. Maybe Ceftriaxone is one way to help them. But I think I got off on a tangent there. But
No, I think that would be great. But might that not work with, I guess, if glutamate is the key to those uncontrolled seizures, it may not matter whether the etiology has something to do with Toxoplasma gondii or it could be another etiology. It might still work, right?
Yes, absolutely.
Yeah.
Mm-hmm.
Because there you're talking about quieting down a particular circuit in the brain that it's responding in a secondary way to the parasite. I guess that would still require some kind of clinical trial, even if you...
unfortunately, a long way off. We don't know how long the effects of Ceftriaxone would be. Do we have to treat, this is a chronic infection that we have no drugs that will remove the source, the cyst. And so do we have to continuously treat with Ceftriaxone, which is really not an option. It'd have to be given IV, for example. And so we have to do many more studies, but it's definitely a patient group that could benefit from this.
Right. And that would be nice.
Yeah.
I mean, to spare young children that kind of disease burden.
Yes. Because they are put on normal anti-seizure medication, but that doesn't always work.
Right. Let me stray a bit from the central nervous system. This parasite is capable of infecting lots of other cells in other parts of the body, right? So does that raise the potential for all kinds of indirect effects? And maybe you're just not focused on that. It's an unfair problem.
Yeah, I mean, the parasite is really amazing. Unlike many other protozoans, it can infect any nucleated mammalian cell on the planet. It absolutely can manipulate many of those. Chronically, in the mouse, we think of the site of infection as being the brain. That is quite localized. But in larger mammals, we, for example, mostly get infected through eating undercooked meat, and that is the muscle of sheep and cows, and
right.
And so there are cysts in muscle cells as well. So there could be a lot going on in the peripheral nervous system that we have no idea about at this point as well.
Right. Well, one reason I was asking that is that I, years and years ago, learned a bit about the nervous system that runs the GI tract.
Yeah. So there's glial cells there too. Uh-huh.
And are there astrocytes? Or?
Yes. Well, I think they're not technically called astrocytes. They're more Schwann cells, and they play a similar role in the peripheral neurons. They're very important physiologically there.
There could be untoward effects of that system that would help to explain how the parasite would, say, disrupt normal activity in the GI tract.
And the GI tract is so linked to the central nervous system. More and more people studying the direct connection, both back and forth. So if you've got infection in the central nervous system, in the spinal cord, for example, people with severe spinal cord injuries have massive issues in the GI tract, and peripheral immune responses as well. So we tend to think of neuroscientists, immunologists, people that work on infection. It's all very closely linked. It's definitely areas in which future studies could focus.
Well, I just wonder if that isn't a good way for us to draw this conversation to a close, because what you've just said is in a sense, is what the future of this field is about, and it certainly you're already showing that the group you're working with has to be interdisciplinary, and you're crossing the boundaries all over the place. But there's more of that to come, it sounds like, with this parasite and with the insights it's providing about the central nervous system. So unless there's some other final thoughts you would like to share, it seems to me this might be an opportunity to let you get on to the important things in your life that have to do with unpacking boxes.
Oh. That's been quite a week or two. But yes, now I'm back at my desk. I get some actual work done. But it was very nice talking to you, Jeff. Thank you very much for your interest as well.
Oh, I think this is wonderful, fascinating area to be working in, and it looks, it's just bound to get more and more complicated.
I totally agree. There's lots of great people working on it, and I think we're just beginning to touch the surface of what's happening.
Yeah, but the insights will come fast and furious, I think. Yeah. So,
our listeners, I think, the parasite is pretty cool. It's pretty good at being able to live in a host for a very long time, and we've got to give it some credit for that as well.
That's for sure. Yeah. Diabolical, but tenacious, and it's too bad it doesn't do nicer things. But
well, if 30% of us are infected, it can't be that bad, right? We're all
Good luck to you, and thanks so much.
Thank you very much.
During this podcast, I spoke with Emma H. Wilson of the University of California, Riverside. I'm Jeffrey Fox of Microbe. Thanks for listening.